This is just my understanding of autism spectrum disorder as an imbalance between the metabolism of the neurotransmitters serotonin and dopamine. Now, if you were to have low serotonin, you would be very aggressive. You'd have very low impulse control. Your thinking speed would race. Your memory wouldn't be that good. Learning wouldn't be that good. Your emotions could be all over the place. High serotonin, you get the inverse. Passivity, great impulse control, slower thinking speeds, improved memory and learning. Same thing happens on the dopamine side. Low dopamine, you lose focus. You're not able to lock onto a train of thought. Your attention level may be low, low excitement, low euphoria. Whereas if it was high, you would focus on things too much. You'd lock on and you couldn't let things go. Your excitement could be all over the place. And similarly, euphoria, the arm flapping. Your average neurotypical person, say, has ratios that fall right here in the middle. And in autism spectrum disorder, in my opinion, it's not a nice straight line. Okay, depending on the ratios, things can go the imbalance can go all over the place. All right, depending on how these two neurotransmitters are working with and against each other. Okay, so an autistic person has imbalances in serotonin and dopamine. Well, they also have intestinal problems in their gut. How is serotonin related to the gut?
Approximately 90% of the human body's total serotonin is located in the enterochromaffin cells in the gut, where it is used to regulate intestinal movements. The remainder is synthesized in serotonergic neurons of the central nervous system and brain, where it has various functions. These include the regulation of mood, appetite, and sleep. Serotonin also has some cognitive functions, including memory and learning. Modulation of serotonin at synapses is thought to be a major action of several classes of pharmacological antidepressants. Serotonin is a neurotransmitter and is found in all bilateral animals, where it mediates good movements and the animal's perception of resource availability. In the simplest animals, resources are equivalent with food. But in advanced animals such as arthropods and vertebrates, resources also can mean social dominance. In response to the perceived abundance or scarcity of resources, the animal's growth, reproduction, or mood may be elevated or lowered. Recent studies involving the serotonin transporter gene 5-HTT have shown the short allele of this gene increases synaptic serotonin levels.
Now, because serotonin in higher life forms is associated with social dominance, you can see how serotonin deficiency would result in social awkwardness and shyness. Makes perfect sense.
Serotonin transporter binding was significantly lowered throughout the brain in autistic individuals compared with controls. Specifically, the reduction in the anterior and posterior cingulate cortices was associated with the impairment of social cognition in the autistic subjects. A significant correlation was also found between repetitive and/or obsessive behavior and interests and the reduction of serotonin transporter binding in the thalamus. In contrast, the dopamine transporter binding was significantly higher in the orbitofrontal cortex of the autistic group. In the orbitofrontal cortex, the dopamine transporter binding was significantly inversely correlated with serotonin transporter binding. Conclusions: the brains of autistic individuals have abnormalities in both serotonin transporter and dopamine transporter binding. The present findings indicate that the gross abnormalities in these neurotransmitter systems may underpin the neurophysiologic mechanism of autism.
So, what causes autism spectrum disorders? It looks like it's fetal vitamin D deficiency in utero during pregnancy. Here's the specific information.
There is growing evidence that low vitamin D impacts adversely on brain development. The current study investigated the impact of developmental vitamin D deficiency on dopamine and serotonin metabolism in the neonatal rat brain. Developmental vitamin D deficiency resulted in an altered dopaminergic metabolic profile in the forebrain, with a decrease in the conversion of dihydroxyphenylacetic acid to amovinoic acid. Correspondingly, expression of the enzyme required for this conversion, catecholomethyltransferase, was decreased. These results suggest that developmental vitamin D deficiency influences dopamine turnover during fetal development.
So if DNA deficiency during fetal development can cause autistic spectrum disorders, can supplementation of vitamin D post-natal, post-birth, maybe mitigate or improve the damage that was initially done?
How does vitamin D help repair DNA damage? According to the investigators in one study, together, our results suggested that activated vitamin D protects cells from insults through inducing DNA repair genes as expression to promote the repair of double-stranded breaks. They went on to say, overall, our efforts support the role of vitamin D in guarding genomic integrity through regulation of genes involved in antioxidation and DNA repair.
So there you have it. That's my understanding of what causes autistic spectrum disorder and how to mitigate the damage postnatally through correction of vitamin D and all the genomic implications that were created during fetal development by the DNA actually repairing itself as best it can. There's some damage that cannot be undone, but I believe much of it can be repaired. Only supplementation and observation will tell the tale.